Hi guys, we are on chapter 44, Osmoregulation and Excretion, and this is our last chapter for the semester. Exciting. So here's how the lecture is going to go. First we're going to talk about osmoregulation, give you some definitions, and talk to you a little bit about the importance of osmoregulation, some of which you've already learned in other chapters. And then different osmoregulatory patterns found in different kinds of animals. And that's what we're going to cover in this first one. In the second, and I don't know if there's going to be a total of 3 or 4 videos, but we'll cover different types of excretion. We're going to talk about different types of excretory organs. And then we're going to spend some time on the vertebrate excretion mechanism, the kidney that you're most familiar with, but that's a pretty complicated process. Okay, so to start out with, osmoregulation is a super important process that all organisms need to partake in because it is the way that we balance how much water we have in our cells, and not only that, but what proportion of dissolved solutes are within our cells as well, because the more water we have, the lower the concentration of solutes. So regulating water is really important for making sure that our organs and tissues function the way they need to. Metabolic reactions depend on certain levels of particular solutes within cells, so we need to maintain that in a type of homeostatic relationship, and osmoregulation allows us to do that. As we learned before, osmosis is the movement of water down its gradient across a selectively permeable membrane. We talked about this when we were talking about plants, and we often talked about it in the terms of water potential. So water moving from an area of high water potential to areas of lower water potential across a membrane. We can also talk about osmoregulation, or an important term in osmoregulation is osmolarity. And so osmolarity is basically a way to discuss the amount of solutes, or the solute concentration of a solution. So how many solutes are dissolved in water. When two solutions are isoosmotic, that means they have the same osmolarity, or in other words, they have the same concentration of solutes. So if you have for example, a cell floating in a medium, like water, and the cell is isoosmotic with the water, that means the water and the cell have the same osmolarity, or the same solute concentration. And then you're not going to have a net movement of water either way because the osmolarity is the same. So that's isoosmotic. However, if the cell or the two sides of a container are not isoosmotic, that means one side, or the cell, is going to be hyperosmotic, and the other side, or the other that's floating in, is going to be hypoosmotic. So these two things, hyperosmotic and hypoosmotic, are basically the way you describe two things that are next to each other, or one thing floating in another, that have different osmolarities. One of them is hyperosmotic and one is hypoosmotic. So the hyperosmotic side has a higher solute concentration and lower free water concentration. That just means that water is not likely to move out of the hyperosmotic side. This will have low water potential, so that's lower free water concentration. The hypoosmotic side, on the other hand, will have a lower solute concentration in general, and that is going to cause high water potential, or higher free water concentration, and the net water flow across a semi-selectively permeable membrane will go from the hypoosmotic side down the water gradient to the hyperosmotic side. and that is osmosis, just using some different terms than what we used before. If we want to think about this in terms of cells and what kind of medium they are in, these are pictures of red blood cells. This one is the way a red blood cell is supposed to be shaped. You see that biconcave shape. And this is what a red blood cell looks like in an isoosmotic medium. Again, that just means that it has the same concentration of solutes within the cell as in the fluid or the plasma that it's floating in. So you do have movement of water into and out of the cell, but it's more or less equivalent. Okay, so you've got isoosmotic medium with the cell, and water can move in and out freely, but there's not a net movement of water into or out of the cell in that case. If, however, the cell happens to be in a hyperosmotic medium, meaning the medium has a higher concentration of solutes, then water is going to move out of the cell and it's going to shrink the cell up. And the opposite happens if the cell happens to be in a hypoosmotic medium, which has fewer solutes in it. And if there are fewer solutes in the medium and the medium is hypoosmotic, then there's going to be a net movement of water into the cell. And you can see that in this case, the cell is expanding to a level that it really shouldn't be. And we need our cells to look like this. So sometimes we have to do some osmoregulation to make sure that water is not moving into or out of our cells at too great of a rate like these. So osmoregulation, as I was saying before, is really important. We have a constant internal environment that we need to maintain, and this is true for all sorts of different things, including pH, salinity, temperature, etc. And this is really crucial to the survival of all cells and all living organisms. And again, it helps us maintain our homeostasis and helps us be able to perform all the cell's metabolic functions. So animals, we've talked about this in plants a little bit, but in animals, we have to maintain constant osmoregulation, or moving water and solutes into and out of cells. We do that typically by moving water. Moving water is a lot easier, but we can also move ions as well. So we have to gain and lose water and ions at appropriate times, sort of back and forth, to maintain this constant osmoregulation or the constant homeostasis. There are several ways that animals can gain water. The main ways are through food and drink, and some aquatic animals also gain and lose water through osmosis across their gills or body surfaces. So we're going to look at this a little bit more when we talk about osmoregulation specifically in fish. But because their gills, for example, are not covered with a protective surface like our skin, water can move in and out of the gills pretty easily. So that's how we gain water, or animals gain water, and animals lose water through the following processes: through producing urine, through producing feces, also by perspiration or sweating, and when we breathe, our breath has a lot of moisture in it, and we lose this from breathing. So there are several different types of osmoregulatory patterns, but they're typically divided into osmoconformers and osmoregulators. Osmoconformers are, by definition, isoosmotic with their surroundings. They are not able to do any kind of osmoregulation, and so they cannot control their osmolarity. This means that their osmolarity is completely dependent on their surroundings, and we're going to talk about that a little bit more in a second. Osmoregulators have a lot more control over their osmolarity, and they expend energy to control how much water they take up and lose in a hyperosmotic or hypoosmotic environment, and different types of organisms are better at those different types of osmoregulation, which we'll talk about in a second. So osmoconformers are found in only marine environments. You cannot be an osmoconformer unless you're in a marine environment. Remember, osmoconformers are isoosmotic with their environment, and most marine invertebrates like crayfish and octopi and mollusks and stuff like that, those are typically osmoconformers. That means they can only live in marine environments. And again, the reason why that is because if they were to live in fresh water, you know that your cells have solutes in them. And if you were an osmoconformer, then basically what would happen is water would rush into your cells and you wouldn't be able to survive. Osmoregulators have a lot more flexibility in where they can live. So almost all vertebrates are osmoregulators. They are either hyper or hypoosmotic with their environment. And terrestrial animals also count as osmoregulators, although they have sort of different problems than aquatic organisms. So we're going to talk about a few of these different examples. Here are some terms that you need to know about osmoregulation patterns. An osmoconformer, as you know, does not change its osmolarity it changes its osmolarity depending on the environment it's in. And it cannot regulate its own osmolarity. So whatever environment it's in, it has the same osmolarity. Here is a graph, a hypothetical graph, listing or explaining what happens to osmoconformers. On the y-axis here, we have osmotic concentration of the blood or hemolymph in the animal. And on the x-axis, we have osmotic concentration of the water. And as you see, for an osmoconformer, as osmolarity increases for water that it's in, so does the osmotic concentration of the blood or hemolymph of the animal. So it's a direct correlation. And that happens in osmoconformers. And this graph right here is called, or this line, is the line of conformity for osmoconformers. Now, what would it look like if we had an osmoregulator? Remember, for osmoregulators, they need to keep their osmolarity at a certain range. It doesn't change much, and it doesn't matter what sort of medium they're in. They have to be able to maintain a particular osmotic concentration of their blood or their hemolymph. So if you want to think about what that would look like on a graph, it would look like this. I mean, probably not exactly like this, but this is representing an osmoregulator who's able to maintain its blood or hemolymph osmotic concentration, no matter what the osmotic concentration of the water was. So osmoregulators would have a much more even line where they maintain a particular osmotic concentration of their blood, no matter what their medium looks like. Osmoconformers change completely depending on the osmotic concentration of the medium. No matter whether you are an osmoconformer or an osmoregulator, most organisms actually have a pretty narrow range of osmolarity that they can exist in and survive. And so those organisms would be considered stenohaline. It means they have a very narrow range of media that they can exist in without dying or while still being able to osmoregulate. However, there are some species that would be considered euryhaline, and those organisms can exist in a wide range of osmolarity. This figure right here, this line, actually would represent something that was a euryhaline, which was able to maintain its same osmotic concentration no matter if it was in purely fresh water or salt water. These species, something like this, doesn't really exist. So again, this is hypothetical. But there are some species who can exist both in freshwater and saltwater. So, for example, mussels or certain invertebrates that may be sort of at the opening of a river that's heading into an ocean. And organisms there have to have an influx of fresh and salt water. So that means the osmolarity of the water changes a whole lot, and those organisms are able to survive that. So that would mean that they were euryhaline. Another example would be fish that spend part of their life in the ocean and part of their life in freshwater. Those organisms, like salmon, for example, also would be considered euryhaline because they can exist in a wide range of osmolarity of the medium that they're swimming in. And those euryhaline organisms tend to have a mixed strategy where they, in some cases, they have to osmoregulate because the water that they are living in is hyperosmotic to them, and they have to use totally different strategies to osmoregulate when the water that they're living in is hypoosmotic to them. So here are some, here's just a figure of some different animals and what sorts of osmolarity their blood has. If we're looking at sharks and rays and marine invertebrates, they are considered more or less osmoconformers. Their osmolarity is equal to that of seawater. So you can see down here, seawater is around 1,000, and that's similar to sharks and rays. But most other organisms, other marine fish, mammals, birds, reptiles, freshwater fish, and freshwater invertebrates, if their osmolarity is not the same as the medium that they're in, then they are osmoregulators. So you can see that a marine teleos, that's just a type, that's a bony fish, they have osmolarity of about 330, but they're living in seawater, which is about 1,000. So they are clearly osmoregulating to keep their osmolarity less than that of seawater. On the other hand, if you're looking at freshwater fish and freshwater invertebrates down here, freshwater has an osmolarity of close to 0, and they have an osmolarity in their bodies of much greater than that. So they've got to osmoregulate in a different way to keep their solutes in their blood higher than the medium that they're swimming around in. So how do they do that? Well, first of all, let's talk for another second about osmoconformers and some of the challenges and some of the benefits that they have. So osmoconformers passively allow body fluids to have a solute concentration that is equal to their surroundings, or equal to the medium in which they are living. Like I said, those have to be saltwater organisms, and they are typically saltwater or marine invertebrates. As I showed you before, osmoconformers follow the line of conformity, but again, they're not found in freshwater, they're only found in saltwater. Being an osmoconformer is actually pretty beneficial because osmoregulators end up having to spend a lot of energy maintaining osmoregulation, whereas osmoconformers can conserve that energy by not having to pump ions in or out of their body. But the disadvantage is that they have a very narrow range of salinities that they can survive in. This is a picture of a brittle star right here, and I'll just tell you this horrible story of when I was a kid and I went to the beach and there was this really cool jetty and I found a brittle star, and my family had a house that was on like a sort of like a river. It was a lagoon and it had brackish water in it. We had some saltwater fish in a fish tank at home using the brackish water from the river or lagoon, and I just figured all salt water was salt water. So I brought my brittle star home from the beach and I stuck him in the aquarium, and I was like, oh look, he's moving around, he's having a great time. Well, what was happening was I put him in an osmotic solution that was hypoosmotic to what he needed to maintain within his cells, and a bunch of water rushed in to this little section right here, and it exploded, and it was very terrifying. But I learned about osmolarity and osmoconformers at a very young age because I had a terrible accident with a brittle star. So some marine invertebrates actually can osmoregulate a little bit. If we were to look at this spider crab line right here, the genus name is Maya, you can see that has a really straight line of osmolarity. And this is a similar graph as before, where you have osmotic concentration of body fluids on the y-axis and osmotic concentration of medium on the x-axis. But the graph is set up a little bit differently, where the saltiest water is on the left and the freshest water is on the right, but everything else is the same. So this guy, Maya, is right along the line of conformity, which makes him a complete osmoconformer. But because these other two marine invertebrates do not have this exact same relationship, looks like they're able to osmoregulate a little bit. They can keep their osmolarity from going down too far, or in fact, keep their osmolarity from going down too far. If they happen to go into fresh water, they can osmoregulate a little bit. Okay, so both of these have some osmoregulatory ability, while this spider crab does not. I'm not sure I'm going to put this now. Okay. So now we're going to talk about problems for osmoregulators. Freshwater organisms are in a hypoosmotic medium. That means when they are swimming around, the water that's around them has a lower solute concentration than their bodies do. So if you want to think about that as these red blood cells that I was showing you earlier, that's like the red blood cell being in a hypoosmotic medium, and water tends to enter the cells or enter the organism. So that's what they have to avoid. That's what freshwater organisms have to avoid. Marine organisms potentially have the opposite problem. Those that are not osmoconformers, they have more salts or solutes in the water outside of the cells than they have in their cells, and water wants to move out of the cells, and so they become dehydrated. So that's marine organisms tend to be in a hyperosmotic medium, where the body fluids have a lower solute concentration, and water tends to leave the cell without osmoregulation. And terrestrial organisms have a similar problem to marine organisms, except for it's not about being necessarily in a hyperosmotic medium as much as it is being in a dry medium, and there's more water concentration inside the cell than outside the cell. So again, water wants to move out for terrestrial organisms. So marine and terrestrial organisms actually have similar problems, even though one lives in salt water and one lives in a terrestrial environment. All freshwater animals are osmoregulators, like I said before. Only saltwater animals can be osmoconformers. So freshwater animals have to maintain their body fluids with solute concentrations different from that of their surroundings. Their body tissue is always in a hypoosmotic environment because they're in fresh water, and they cannot survive at an isotonic or isoosmotic concentration. That would mean all their solutes would basically leave their body. And they have to avoid the problem of preventing water into their body. And I know I'm repeating a lot of these things a bunch, but this is kind of confusing, so I just want to make sure that it's getting across. So if we talk specifically about freshwater fish, they have particular ways of combating this problem. Again, the problem is that they are in a hypoosmotic medium, and water tends to rush into their body because of that. Also, they tend to lose solutes by diffusion out of their gills, so that's not good. They gain water and they lose solutes just by being in fresh water. So the ways that they combat this, the ways that they osmoregulate, is that they do not drink water because they don't need more water. The water that they need to get is coming in through their gills. They pump solutes into the gills. Actively, solutes are leaving the gills, so they have to passively, solutes are leaving the gills. So they have to actively pump solutes back in to the gills. And they also release a large volume of dilute urine, and that's one way of getting rid of some of that excess water. Saltwater fish have the opposite problem. Their body fluids have lower solute concentration than their environment, and they are in a hyperosmotic medium. So again, that's like with a bunch of solutes in the outside, and you tend to lose water. So the problem is that they lose water by osmosis, and solutes tend to actually come into their gills passively through diffusion. Again, that's just because there's a bunch of solutes in the water and they move passively into the fish. So the solutions that saltwater fish have, if they are not osmoregulators, is they drink a lot of seawater, so they're gaining a bunch of extra seawater or extra water through drinking, and they pump out solutes by in their gills. So their gills have these cells that pump out solutes, and in addition, they don't release very much urine at all, and it's very, very concentrated. Terrestrial organisms, like I said before, they have similar problems to saltwater fish or saltwater organisms that are in a hyperosmotic environment. So they have an impermeable covering, which reduces water loss, but they still do lose water through evaporation from respiratory surfaces, like when you breathe. Also body surfaces from perspiration, feces, and urine. So these are all the same ways I mentioned before that we can lose water. And so they have to conserve water, and they also have to consume extra water. So they get additional water by drinking or eating. Also, they can get some water by as a byproduct of some of their metabolic processes. So as you know, cellular respiration has water as a byproduct. Also, they have to excrete, they have to minimize water loss in their excretion. So excreting ammonia would be energetically efficient. We'll talk about this a little bit later, but it takes way too much water to dilute it because ammonia is toxic. So what terrestrial animals do is they excrete urea, which doesn't require quite as much water to reduce the toxicity. And so that's one way that they can minimize the amount of water that they lose in excretion. Okay, so that is it for the first lecture. And I will talk about excretion and maybe get into the different types of excretory organs in the second one.